Ever wondered how a butterfly emerges from a caterpillar? Or how a tree grows from a small seed? How do we get energy? What happens inside our body? How do we fight diseases? All of us are curious about such and many other things, including broader questions about life around us. Curiosity is one thing that drives the researchers at the Indian Institute of Technology, Bombay, to explore and investigate the biological world and to seek answers to fundamental questions in biology. Life begins as an embryo, which is just a single cell. In the Vertebrate Embryogenesis laboratory, Professor Sreelaja Nair studies how an embryo grows into an organism.
From that one cell, a diversity of cell types have to emerge: the brain, skin, eyes. That single cell is a totipotent cell, which means it can give rise to everything. But over time, cells have to decide that they are going to make the brain or the lung or the liver. That's called cell fate specification.
Once fate is specified, cells of a particular fate move from their place to come together and form an organ.
A major focus of my group is to try and understand how cell fates are assigned during early development of zebrafish embryos, and how do these cells move from one location to the other, where they will then differentiate to give rise to different types of organs.
The embryo is about 10 times larger than most cells in the body. It divides and makes cells that are much smaller in size.
Not only the cell has to shrink in size, whatever is inside the cell, the nucleus, the mitochondria, the Golgi, endoplasmic reticulum, many of these fundamental cellular organelles also have to shrink in size. And we try to understand, when a cell is large and it has to transition into a small cell, how does it scale? Is everything linear, or does something scale out of proportion with each other? We use zebrafish as a model organism. They share 70% of genes with humans. It's a vertebrate. It's a small model organism. It's easily maintained in the lab, and a single pair mating can lay thousands of eggs. The embryos are completely transparent, so you can watch organs, cells move in real time in a live embryo from the beginning till the organ is fully formed.
Spinal muscular atrophy is a disorder which affects the spinal cord nerve cells that control muscle movements. As a result, muscles shrink and stop working. If not treated, infants with spinal muscular atrophy are likely to live for just a couple of years. Therapies are few and prohibitively expensive. Professor Nair and team are trying to understand what causes spinal muscular atrophy. They are looking to develop a proof of concept for a gene therapy-based potential cure for spinal muscular atrophy. They plan to mimic spinal muscular atrophy causing mutations in zebrafish. They will then inject human cells that have the corrected gene and observe if these cells can correct the zebrafish genes and thus cure the disease.
The reason we are excited about this is because what this quite easily gives us is a proof of principle concept that a human genetic disorder can be corrected using basic biology techniques in a model organism.
In the motor protein laboratory, Professor Roop Mallik and his team study motor proteins, tiny machines that move cell components from one place to the other inside the cell.
We try to understand how these motor proteins generate force, talk to each other, how they are controlled by your brain or by different parts in your body so that your metabolism can work well.
Motor proteins are nano-sized, about a millionth part of a millimeter. They exert forces in few piconewtons, about 1,000 billionth of the force we exert when we push a shopping cart.
We can also measure this force, which we do by shining a laser beam inside a cell. Let's say a bacteria which is happily moving around trying to survive. We can shoot a laser beam on this bacteria. When the laser beam focuses onto the bacteria, you can actually exert a force because of the light that you are shining, and we can then measure how much force is being exerted by these motors to pull these bacteria around, and from this we can infer how the activity of these motors is manipulated by the bacteria to achieve a desired result.
Professor Mallik is studying the role of motor proteins in how pathogens attack a human cell and how the cell tries to kill the pathogen inside the cell. The pathogen hijacks the cell machinery and uses the motor proteins to move to a place in the cell where it can replicate. The host cell encloses the pathogen in a chamber. Motor proteins called dynein push the pathogen chamber to an acidic chamber where the pathogen is killed.
In this context, we found out that cholesterol, which all of us don't like, is actually the lipid that might be helping you kill the pathogens.
Cholesterol in the cell forms platforms over the pathogen-containing chamber, helping dyneins to accumulate on it.
The other aspect is how your body manages fat. Your liver is the central organ in your body which decides how much fat is going to be there in your blood. There is a tiny motor called the kinesin motor. This kinesin motor is moving very tiny fat particles inside the cells of your liver to a location where these fat particles can be digested and then sent out into your blood.
If the lipid particles can be blocked in the liver and prevented from entering the bloodstream, it can help patients who have problems associated with high blood cholesterol. Professor Mallik is also studying the interaction of antidepressant and anti-cancer drugs on the balance of lipids in the body. Professor Swapnil Shinde and his team at the Cilia Biology Laboratory study primary cilium, a cell organelle that plays a crucial role in cell signaling or the communication that governs basic cellular actions.
There are two types of cilia. One is motile cilia, the ones which are found in our airway epithelia, where they beat in synchrony to move the liquid or fluid. Then these motile cilia are found on small biflagellates or freshwater algae called Chlamydomonas, where it uses it for the swimming. But the cilia is called as the primary cilia. It is non-motile, a single solitary structure present on the surface of the cell.
Cilia in our sensory organs help us sense through special receptors called GPCRs or G-protein coupled receptors. Cilia do not make their own receptors or proteins. Every receptor and signaling molecule that functions in the cilia is synthesized in the cell and transported to the cilia by a specialized machinery. Once their job is done, the proteins and receptors are removed from the cilia by another specialized machinery, which also removes proteins which may land in the cilia by mistake.
The machinery called the ribosome, it kind of acts as a vacuum cleaner to maintain the quality control into the cilia. The question is, how does this retrieval complex recognize that these proteins have to be removed? We identify a mechanism where we showed that when these G protein coupled receptors which are present on the cilia, when they get activated, they get ubiquitinated, and this ubiquitin kind of flags to the system that this receptor is activated and it has to be removed out of the cilium, whereas the other protein should be left in the cilium. The next question we asked is that how does the retrieval machinery knows that this is the ubiquitin that I have to go and bind to? We employed a proteomics approach where we tried and identify the ubiquitin readers which can go and specifically recognize these ubiquitinated proteins. At the same time, this ubiquitin reader can go and bind to the retrieval machinery.
Any disruption or defect in the function of the cilia causes a group of diseases called ciliopathies. Ciliopathies are multisystemic, which means that a patient can have polycystic kidney, retinal degeneration, craniofacial defects, and congenital defects too.
There is no cure for ciliopathies, and our current understanding of molecular basis for the ciliopathies is limiting. You understand the molecular basis of these diseases, that's another aspect that we are looking into, how several G protein coupled receptors play important role to regulate body homeostasis.
The path led by curiosity helps discover the fundamental mechanisms underlying the fascinating world of biology. As researchers at IIT Bombay get deeper insights into how embryos develop, how components move inside cells, and how essential messages are exchanged in the cells, they uncover secrets that tell us how our bodies work in health and disease.